Hi, my name is Bohdana Doskaliuk, and I'm an associate professor at the Ivano-Frankivsk National Medical University. Today, we will talk with you about the nanomaterials in drug delivery systems, from their concepts to the practice use. I would like to disclose that I do not have any conflicts of interest. So, what is a drug delivery system? Drug delivery system, or DDS, is broadly defined as a formulation or device that enables the introduction of a therapeutic substance into the body and its transport to the target area, potentially enhancing the drug's efficacy and safety. In other words, DDS controls how and where a drug is released in the body, aiming to maximize therapeutic benefit while minimizing side effects. Modern DDS technologies include nanocarriers, tiny particles on the nanoscales, often in between 1 to 100 nanometers. They can carry drugs and release them in a controlled manner. Why do we need a specialized carriers devices? First of all, let's go fast through the challenges of so-called free drug molecules. Many drugs have a poor water solubility. It means low bioavailability for the organism. Also, most of the drugs might have some fragile molecules like proteins or RNA. They can degrade before reaching the target. How the nanoparticles can help us to resolve these problems? The encapsulation protects drugs from the degradation or inactivation beforehand. Improves solubility of hydrophobic drugs, prolongs circulation time in the bloodstream, so it means better tissue accumulation. They also enable targeted delivery, so more drug comes to the diseased cells, less to healthy ones. Nanoparticles also allow controlled release of the drug in response to the pH or temperature. So what are the clinical benefits? Basically, nanoparticles enhance stability and improves pharmacokinetics. Lower doses and fewer side effects are also done by the nanoparticles. On this slide, you can observe the most important advantages of nanomedicine. So here we can see that the small size, the different shape, the charge on the surface of nanoparticle, the possible functionalization, combination of different drugs, even the triggered release of the drug is all possible thanks to the nanomedicine. So what are the key requirements for DDS? First of all, biocompatibility. The delivery system should not be toxic or trigger a harmful immune response. In fact, avoiding detection and clearance by the immune system is one of the biggest challenges in developing nanomedicine. The materials used should be safe and ideally biodegradable to prevent long-term accumulation. The DDS should provide controlled release of the drug, for example, slowly releasing the payload to maintain a therapeutic level, or release it in response to some particular trigger. It also should achieve targeting, either by passive means or active ones. Classification of nanomaterials for drug delivery includes various classes of materials, generally categorized into organic, inorganic, and hybrid nanocarriers. Each class has unique structures and properties that can be leveraged for delivering of the drugs. Let's start talking from the lipid-based nanoparticles. These are the small spherical vesicles that are versatile nanocarriers. They are made of one or more phospholipid bilayers, which are similar to the cell membranes. They are biocompatible and biodegradable. They can carry both hydrophilic and hydrophobic drugs. The hydrophilic drugs will be stored in the aqueous core, and the hydrophobic will be embedded in the lipid membrane. From the advantages of this method, The lipid nanoparticles can protect drugs from degradation, they reduce toxicity to the organism, and they enable controlled release triggered by heat, light, or magnetic fields. We do have numerous clinical successes with the lipid-based nanoparticles. For example, the Doxil liposomal doxorubicin. This is the first FDA-approved nanodrug back in 1995. It is PEG-coated for immune evasion and tumor-targeting nanoparticle. It has reduced the cardiac toxicity in comparison to the free doxorubicin and is still used in the cancer therapy. However, we cannot forget about the possible disadvantages of the lipid-based nanoparticles. First of all, it's their physical and chemical instability. There can be the aggregation or leakage of drug during the storage. They are sensitive to temperature, pH, and especially lipid peroxidation. Also, the lipid-based nanoparticles do have the limited drug loading capacity. They can be rapidly cleared from the organism by the mononuclear phagocyte system. Also, there are the challenges in the large-scale production of those nanocarriers. And we do have the concerns about cost and complexity of production. What about the polymeric nanoparticles? What are they? They are formed from the biodegradable polymers. They can be synthetic, like PLA, PGA, PLGA, or natural, as chitosan or alginate. There are two main types of them. Nanocapsules, core shell with the drug in the core, or nanospheres, solid matrix with the drug evenly dispersed. What are the key advantages, you will ask? So first of all, it's the tunable structure, which ensures a controlled drug release, good stability in biological environment, biodegradability, and also the polymeric nanoparticles can protect the drug from degradation before the release. It has numerous applications like delivery of chemotherapeutic drugs, proteins, or even genes. We do have also the ability to produce the inorganic nanocarriers. So they mostly are metals, metal oxides, or silica. These nanocarriers, nanoparticles, tend to have very defined and stable structures. A prominent example is the gold nanoparticle. It can be made in various shapes: spheres, rods, shells. Also, there can be different sizes. And what is the most important, gold nanoparticles has the unique optical properties. Gold is also comparatively inert. Gold nanoparticles are generally low in reactivity and toxicity and can be made biocompatible. Another big advantage is the ease of surface functionalization. Gold surface binds really good to the molecules containing thiol or amine groups, so one can attach drugs targeting ligands or polymers onto the gold nanoparticles. This allows gold nanocarriers to carry a wide array of cargo, the chemotherapeutic drugs, proteins, DNA, RNA, or to be coated for stealth and improved circulation in the organism. The one challenge with the inorganic particles like gold is that being non-degradable, they may persist in the organism for a pretty long time. Researchers often mitigate these nanocarriers by limiting their size so they can be excreted by the kidneys. There is also the group of magnetic nanoparticles. They are typically made of iron oxide. These particles have the spherical property of special property of responding to magnetic fields. In medicine, They can be used for magnetic targeting. Magnet can be used externally to guide the particles to a specific location in the body or to hold them at a site for localized treatment. Moreover, an alternating magnetic field can heat up magnetic nanoparticles, which can trigger drug release or even kill cancer cells by heat. Iron oxide nanoparticles are also valued as MRI contrast agents due to their magnetic properties. So, magnetic nanocarriers serve dual roles: drug delivery and imaging. So, that came under the common name as theranostic, about which we shortly will be talking on the next slides. Silica nanoparticles. Silica, or silicium dioxide, can form nanoparticles that have a honeycomb-like porous structure. Mesoporous silica particles contain an organized network of tiny pores, typically from 2 to 10 nanometers, in which drug molecules can be loaded. They act like the sponges carrying the drugs. The pores can be capped or gated with the stimuli-responsive molecules, enabling the controlled and on-demand release of the drug. The mesoporous silica nanoparticles are attractive for the researchers and the doctors because of their high surface area and pore volume. So their capacity of the drug loading is pretty high. Also, the silica carriers have been tested for the delivering of the cancer drugs, genes, and even for imaging purposes. The one consideration is, though, that pure silica is not biodegradable in the body. However, if the particles are small enough or the body can excrete them, they can be used safely. Research is also ongoing into the silica particles that dissolve slowly in the bodily fluids. We have also the hybrid and composite nanocarriers. Hybrid nanocarriers refer to systems that combine multiple types of materials to leverage the advantages of each. For example, a common strategy is making lipid polymer hybrid nanoparticle, where a biodegradable polymer core, providing structural integrity and controlled release, is coated with a lipid or liposome outer layer, which provides the biocompatibility and a natural interface with the body. These hybrid nanoparticles can improve drug loading and stability compared to liposomes alone, while still remaining biocompatible. Another hybrid approach is inorganic organic composites. For instance, embedding magnetic or gold nanoparticles inside a polymer or liposome. This way, one nanoparticle can both carry a drug and respond to an external stimuli or serve as the contrast agent. Such multifunctional nanoplatforms are the basis of theranostics, so they combine therapy and diagnostic in one. A classic example is a magnetic luminescent nanoparticle coated with the targeting molecules and loaded with a drug. It can be tracked by MRI or optical imaging, guided to the tumor, and then triggered by a magnetic field or light to release the drug. Researchers have also developed stimuli-responsive hybrids. For example, iron oxide core with a heat-sensitive polymer shell that releases drug upon heating. On this slide, you can observe the overview of typical groups and types of organic nanocarriers. Here you can look through summarized strength, weaknesses, their main features, biocompatibility, and even clinical use. On this slide, you have the four main group of the inorganic nanocarriers and also their main features, clinical use areas, their biocompatibility level, and, which is maybe most important, their strength and weaknesses. So what are the principles of targeted drug delivery? One of the most powerful features is the ability to target drugs to specific tissue or cell. There are two main targeting strategies in the nanomedicine. The passive targeting, which works with the enhanced permeability and retention effect, EPR, or active targeting, ligand-receptor interaction. The passive targeting exploits natural physiological differences between healthy and diseased tissue. In cancers, for example, tumors often have leaky blood vessels and poorly functioning lymphatic drainage. This phenomenon, known as EPR effect, allows nanoparticles, typically between 50 to 200 nanometers, to extravasate from the bloodstream into the tumor tissue and then remain trapped there, since the impaired lymphatic tissue cannot effectively clear them. As the result, nanoparticles tend to accumulate in many tumors much more than they would in the normal tissue. On the other hand, the active targeting involves adding a specific homing mechanism to the nanoparticle. This is typically done by attaching targeting ligands to the surface of the nanocarrier. These ligands can be antibodies or antibody fragments, peptides, small molecules, or other biomolecules that selectively bind to receptors or antigen overexpressed on the target cells. The end result is the higher amount of drug entering the disease cells compared to nanoparticles without the targeting ligand. On this slide, you can visualize what are the main differences between the passive and active targeting in our tissues. So, controlled drug release. What is that and how it can be achieved? Controlling when, where, and how much drug is released from the carrier is the crucial aspect of DDS design. Unlike a conventional pill or injection where the drug is released immediately and often throughout the body, nanoparticle systems can be engineered to release their payload of the drug in a controlled fashion. For instance, slowly over time or only in response to certain stimuli. Controlled release helps in maintaining effective drug concentrations, reducing dosing frequency, and targeting drug actions to specific sites. What can be the stimuli that are used to release the drug from the nanoparticles? First of all, it can be the pH. For example, the tumors and inflamed tissues are more acidic than the normal blood. So we can use the pH-sensitive nanoparticles that will remain stable in the bloodstream, but will quickly destabilize in the acidic environments. We can trigger the release of the drug also with the temperature. The thermosensitive carriers release drug when heated for example, 42 degrees of Celsius. Also, we can use the external physical triggers like light-triggered systems, magnetic field, and even ultrasound. There are a few mechanisms of drug release. First is the diffusion. It's the simplest mechanism. The drug slowly diffuses out of the nanoparticle matrix or core into the surrounding environment. Many polymeric matrix systems work this way. The drug is mixed in the polymer, and as water penetrates the nanoparticle, the drug molecules diffuse out. Degradation or erosion of the carrier is also pretty suitable, especially if we want to release the drug over time. So what is it? In biodegradable systems, the nanoparticle itself will break down over the time, either by hydrolysis or by the enzymes present in the body. As the carrier degrades, it releases the drug. As we were talking before, also we have the stimuli response release. As the nanoparticle will be affected by the change of the pH or light or magnetic field, the drug will be able to be released in the tissue, and we will have the pretty much targeted effect on the cells that should be treated. In many nanoparticles, this mechanism of the drug release act in combination. For instance, in a PLGA nanoparticle, there might be an initial diffusion or surface accessible drug, then slower release as the polymer matrix erodes. It will be the combination of a diffusion and a degradation. In, for example, a mesoporous silica, some drugs might release by diffusion until the gatekeeper molecules are reached. These gatekeeper molecules will then open by some particular stimuli to let out the rest of the drug. So this is called the hybrid controlled mechanism of the drug release. Let's talk about the delivery routes and the target sites for the nanoparticles. As you may have already understood, most of the nanoparticle-based drug delivery systems have been researched and implemented into the cancer treatment. So on this slide, you can use the QR code to get more familiar with this process. Another important use of the nanocarriers is their abilities to cross the blood-brain barrier. You can see that due to their small size, they can use the transcytosis or paracellular transport as the option to cross the specific barrier of our organism. It's very important to understand that the cancer treatment is not the only one area where we can use the nanoparticles. The recent advantages in biomaterials have led to modifications that enhance bone cement's therapeutic potential. These innovations can actively influence the bone healing microenvironment through tailored physicochemical properties. We can use the nanomaterials and nanocarriers to actually improve the angiogenesis or regeneration and immunomodulation in the bones or other soft tissues by the targeted influence. To enhance the delivery of some of the drugs, like in this case quercetin, it was encapsulated in the phenylboronic acid modified dendrimer nanoparticle and embedded in a ROS responsive injectable hydrogel for targeted on-demand release. It was the study of the Chinese colleagues that have tried to decrease the intervertebral disc degeneration with the help of such method. On this slide, you can see that actually this quercetin hydrogel method can mitigate the IDD progression. Another interesting way to use the nanocarriers is to affect the eye tissue. So previously, the cataract was not treated effectively by the therapeutic drugs, mostly only by the surgery. But the innovative delivery platform offers a promising and non-invasive strategy for treating or preventing anterior eye segment diseases, even including the cataract. Even in the development of our immunity against the different infective diseases, we can use the nanocarriers as the helping hand. For example, the novel adjuvant delivery system constructed by aluminum emulsion hybrid nanoparticles can boost the vaccine immunity, so that also can be pretty handy. despite we have such a huge variety of the nanoparticles, still some problems in the penetration of the blood-brain barrier or other specific barriers in our organism remains. To address these limitations, Zhang et al. Proposed a groundbreaking approach back in 2011. So his idea was to develop the cell membrane-coated nanoparticles. This biomimetic platform involves wrapping synthetic nanoparticle core with the natural plasma membranes derived from the cells, such as RBC, leukocytes, or stem cells, which can be isolated from peripheral blood using standard techniques. These cell membrane-coated nanoparticles replicate key biological functions of their source cells, including immune evasion, tissue-specific targeting, and improved transendothelial migration. By enhancing both as systemic circulation of nanoparticle as blood-brain barrier permeability, this strategy represents a major advancement in the development of nanotherapeutic drugs for CNS disorders. So here you can see the scheme of the technique for the producing cell membrane-coated nanoparticles. To fabricate them, you can first harvest the cells that you need from the organism of the patient, and then these membranes will be applied to the nanoparticle cores using the three main approaches. It can be done with the mechanical extrusion, ultrasonic sonication, or microfluidic electroporation. Here we can see the schematic visualization of how the specific neutrophil membrane-coated nanoparticle can interact with the blood-brain barrier and other cells and actually deliver the drug to the site of the problem. Till now, we do have a lot of success stories in the nanoparticle therapeutics in the clinics. So the first nanodrug to receive the FDA approval was Doskaliuk in 1995, so pretty long time ago. Doskaliuk is the pegylated liposomal formulation of doxorubicin, used initially for Kaposi sarcoma and later for ovarian and cancer and multiple myeloma. Clinically, Doskaliuk demonstrated how nanocarriers can improve a drug's performance. Patients were having less cardiotoxicity compared to the regular doxorubicin. It all was because of the liposomes shielded the heart from the exposure, and the drug mostly was concentrating in the tumor. Not long after, other nanoparticle drugs followed. We can also talk briefly about the Abraxane because it uses a little bit different technique. It is actually the albumin particles that are carrying the paclitaxel. They are increasing the paclitaxel solubility and tumor delivery, and crucially, it avoids the need for toxic solvent chemophore, thereby reducing hypersensitivity reactions and side effects in patients. There are the numerous cases of approved nanoparticles in the drug delivery systems. We can just highlight a few of them, like the liposomal irinotecan or liposomal daunorubicin. Also, we have the pegylated substances and even RNA lipid nanoparticles. Also, there are the group of antibody-drug conjugates. Sometimes they are considered separately from the nanoparticles, but they embody the targeting principle as well. So from lab to the clinic is the really very long path. We all need to understand this. To develop a drug delivery system with a nanoparticle that is ready for the clinical use is really a complicated task. One challenge is the extensive toxicity testing, Because of nanocarriers often have very different biodistribution and metabolism compared to the usual molecules, they must undergo careful evaluation on animal models to ensure they do not cause unexpected harm. Nanoparticles have large surface areas and can interact with the biological components in unpredictable way. For example, they can boost the immune system too much, or they can activate the complement system. Also, we need to ensure the biocompatibility of the nanocarrier. They should ideally be inert in our organism. Also, it's very important that after the delivering of the drug, the carrier should either break down into harmless metabolites or be excreted from the body. If not, there is a risk of accumulation in organs like the liver, spleen, or lungs. Persistent particles can cause long-term toxicity. For example, certain inorganic particles might generate reactive oxygen species or interfere with the cellular processes if they are accumulating too much. Regulators that need to approve the drug will ask for studies on what happens to the nanoparticle compounds in the body over time. Do they fully clear, and by what route? And this is the part of understanding the pharmacokinetics of the drug delivery system, not just the drug itself. Another complications on the path to being approved, it can be the manufacturing complexity. Also, the characterization requirements, like the nanocarriers should have the same size, surface charge. They need to have the sterility and batch-to-batch consistency. And yes, the regulatory standards of the FDA and other standards commissions are evolving each day. So there are all the time some novelties. We cannot forget also about the cost and trial design, the production and storage of the nanocarriers is pretty expensive. Also, we do require the strong collaboration between the academia, between the industry and the regulators, and only in that case we can achieve the streamlined development of these products. Here on this slide, you can actually observe the potential risks and considerations for the nanoparticle use in the organism. We can see how they are delivered into our body on the systemic level, how they can cross the biological barriers, what they can do on the tissue level, and even on the cellular level in our body. So, among the previous considerations, we need to point out the non-specific interactions. So the blood proteins rapidly form a protein corona, altering nanoparticle behavior. They can lead to off-target uptake and aggregation. About the accumulation in the organs, we need to highlight that if we are choosing the small nanoparticles, less than 6 nanometers, they can be cleared with the kidneys through the urination, but if they are from 10 to 100 nanometers, they are prone to organ accumulation. Some materials may cause inflammation or even granulomas due to their material-associated toxicity. Even biodegradable carriers may have the toxic monomers or residues. Also, we need to think about the disposal of nanomedicines, because there are also the environmental aspects of this area of studies. So, to sum up, nanoparticle drug delivery systems hold great promise and have achieved clinical success. However, careful risk-benefit analysis is crucial for their development. The trend in recent years is encouraging. More nanomedicines are entering clinical trials than ever before, including in cancer, infectious diseases, and genetic disorders. Each new system must prove that it offers a significant advantage in comparison to the traditional drug to justify any new risks or costs. With advancing technology, better characterization tools, and growing regulatory experience, the pipeline of nanotherapeutics is likely to expand, bringing more innovative treatments to patients in the future. Thank you for your attention.